Today, in part 8 of our unit on heredity, we're going to discuss material from pages 236 to 239. Specifically, we're going to talk about the important work of Thomas Hunt Morgan and his famous experiments on the common fruit fly. Drosophila is the genus name, the scientific name for the common fruit fly, and that was the organism of choice for T.H. Morgan as he sought to understand how heredity works. Now, T.H. Morgan began his career in biology in the late 1800s as an embryologist. He was interested in the development of embryos. That was his specialty. Because the scientific field of genetics really did not exist at that point. Remember, Gregor Mendel's work, which was published in 1865, had been forgotten about, and no one really understood it. But in 1900, right at the beginning of the 20th century, Gregor Mendel's work was rediscovered, and finally it was appreciated by the scientific community, and a whole lot of biologists became really excited about this exciting new field of genetics. And one of those people who got really excited about it was T.H. Morgan. I think it's safe to say that Morgan is the second most important geneticist of all time, after, of course, Gregor Mendel. And one of the things for which T.H. Morgan is famous is a discovery that he made in 1910 when he became the first scientist ever to be able to associate a specific gene with a specific chromosome. And what I mean by that, you have to realize in the early 1900s, even though people were starting to understand how genes work, and people were starting to understand Gregor Mendel's rules that explain how genes work, nobody knew yet, including Gregor Mendel. He also never knew what a gene actually is. And this was very shortly after the discovery of chromosomes. Chromosomes were discovered in the early 1900s. And people started to realize that maybe genes, whatever genes are, maybe they are somehow attached to or carried on chromosomes. And they made that connection between genes and chromosomes. In fact, that became known as the chromosome theory of inheritance, and T.H. Morgan was one of the early proponents of that. But he took it a step further. He was able to figure out exactly which chromosome carried a particular gene in fruit flies, and we'll talk a little bit about that famous experiment that took place in 1910. Now, you might wonder, why would a scientist choose fruit flies as an organism to experiment on? It does seem like a bit of an odd choice. First of all, you may not even realize exactly what we're talking about. What is a fruit fly? You've probably seen them, but you may not have realized what you were seeing. Fruit flies are much, much, much smaller than, say, a common house fly. They're really more about the size of a gnat. So if you've ever had some apples or bananas or even onions from the garden that sit out at room temperature too long, maybe for several days, and they start to get a little bit soft and they're starting to go bad, sometimes you'll notice what seems to be little gnats flying around that fruit or the vegetables. And those are Drosophila, those are fruit flies. And the reason they hover around soft, rotting fruit is because they lay their eggs, the females lay their eggs in that fruit or onions or whatever it is. And that way, when the eggs hatch, the baby flies, the maggots, will have something on which to feed. They actually will feast on that rotting fruit. So it's a strange little organism and very tiny, but there are lots of good reasons why this organism is a good one to use for genetics. First of all, they are very small and easily maintained. Um, you can, and I've done this in college genetics labs, you can grow hundreds and hundreds and really thousands of fruit flies in a very, very, very small container. Um, you, Little containers maybe about the size of even smaller than maybe a television remote, or maybe about the size of a marker that I would use to write on the whiteboard. You can grow hundreds of flies in a tiny little vial. That's how small they are. So, and all you have to feed them is some moist potato. I know we always used instant mashed potatoes. We put a little bit of mashed potato powder in the bottom of the container, a little bit of water, and that's it. That's all they need. They get all the food from that. They have a very short life cycle. So from the time the female lays her eggs until they form a pupal case, and then eventually a maggot hatches out of that, and then it forms another pupal case, and then eventually the adult emerges. From the time the female lays her egg until that egg grows up to be an adult fly ready to lay its own eggs is less than 14 days. It's usually more like 11 or 12 days. So that's really fast. Now compare that to Gregor Mendel. When Mendel crossed two pea plants, when he cross-pollinated those pea plants, he had to wait months for the seeds to form. Then he had to pick those seeds. Then he had to dry them out. Then he had to wait until the next spring to plant them. So you're talking a year or two just to get a couple of generations of pea plant data. You could get that same amount of data from a fruit fly cross in 2 to 4 weeks. So it's much faster. The generational time from one generation to the next is much, much faster with fruit flies than it is for many, many other organisms. Genetically speaking, fruit flies are pretty simple. They only have 4 pairs of chromosomes, so that's 8 chromosomes total, and they have 1 pair of sex chromosomes, just like us humans. And even the way gender is determined is the same for fruit flies and humans. So XX is a female fruit fly, XY is a male. So lots of reasons why Drosophila make a really good choice. And in fact, I can tell you that at the college level, every genetics experiment I ever was a part of used fruit flies. They're very commonly used in genetic research even today. But that all started in the early 1900s with the work of T.H. Morgan. So I mentioned this already. Here's a male and a female fruit fly. So when they mate and the eggs get fertilized, the female, of course, lays the eggs. About 1 day later, a little tiny maggot hatches out of that egg and starts to feed on the rotting fruit. And after just a few days, that maggot is gotten pretty big, and it forms a pupal case, which is basically like a little tiny maggot cocoon. And then about 4 days later, it hatches into an adult. So the whole process is maybe 11 days. It depends on temperature. Sometimes it can take as many as 14. So that's really fast. It's way faster than pea plants. But just like pea plants, fruit flies have a lot of variety. Your common typical fruit fly has kind of a tan-colored body with black stripes and red eyes, and of course 1 pair of wings to fly. But there are all kinds of different traits that T.H. Morgan identified. So there's different eye colors that some fruit flies have. Some of them have a darker body color. Some of them have wings that are really, really small, so small that the fly cannot actually fly. They're called vestigial wings. So there's all these different traits that are genetic traits, and that's what T.H. Morgan was interested in, is how are these different eye colors and body colors and wing shapes, how are those inherited, and how are they passed down, from one generation to the next? Very much like Mendel, was studying how traits are passed down through the generations of pea plants. So you might wonder, when you're working with these very tiny little insects, how can you even tell which ones are male and which ones are female, if you're trying to get them to mate? And it's actually pretty easy to do that. There are very simple methods. I won't go into a lot of detail here, but there's a way to anesthetize the flies. So you literally temporarily put them to sleep with a chemical, and then you can lay them out on your lab bench and use a magnifying glass or a simple microscope to magnify them so you can look at them and you can tell what color their eyes are and so forth. But you can also tell which ones are male and which ones are female. If it's a male, there's going to be a big black stripe on the abdomen, whereas the female has more narrow, skinny little stripes. Also, the male's body is a little smaller, and the females have a little pointed tip right here on the tip of the abdomen. That's a little structure called an ovipositor that she uses to lay her eggs. And the male obviously doesn't have that because he's not going to lay any eggs. So that's how you tell them apart. Let's see. Well, let's go ahead and talk about this, I guess. Since we're talking about the work of T.H. Morgan, it's important that you understand his system that he and his students devised for representing alleles. For some reason, they chose not to use the traditional system of taking the letter of the dominant trait and using a capital letter for the dominant allele and a lowercase letter for the recessive. I don't know why they didn't stick with that, but they came up with their own little weird system, and this is still used today. Whenever you're doing fruit fly genetics, we still use T.H. Morgan's system for symbolizing genes. So what he would do is he would study and he would inspect hundreds and thousands of fruit flies until he found one that looked different in some way or unique in some way. Maybe it had white eyes, even though 99.9% of fruit flies have red eyes. Or maybe it had a really dark colored body, even though 99.9% of fruit flies have that light colored body. And whenever he found that oddball, he figured, well, that's got to be a mutation. There must be a new allele that was created by a mutation that caused that fly to look different. And so he would name the new recessive allele using letters from that particular trait. So, for example, let me see if I've got a picture. Yeah, so look at this fruit fly. Notice instead of normal wings, it has these little wrinkled up, not fully formed wings. It looks like something is clearly different about the wings of this fly. This fly has wings that are not flight capable. They're called vestigial wings. They're non-functional. Obviously, a fly like this would not survive very long in the wild because it wouldn't be able to go search for food. But in captivity, where the food is plentiful, they survive just fine, and they can pass down that vestigial gene to the next generation. So anyhow, when he discovered this mutation, he named it VG. And so he's using either the first letter or, in this case, a combination of two letters to represent that mutation. So any fly that is VGVG, that's homozygous recessive, that fly is going to look like this. It's going to have vestigial wings. If you put a plus on the VG, that little plus indicates that the allele is the wild type allele. Now that's kind of a strange term. In genetics, wild type means if you were to look at thousands and thousands and thousands and thousands of these flies in nature, in the wild, most of them would look a certain way. Most of them would have normal flight capable wings. So we call that wild type. Most of them would have red eyes. So that's the wild type eye color. And so that's what that means. It's sort of like the normal phenotype that you would expect to see in the wild. And in this case, because vestigial wings is recessive, VG is the recessive allele for vestigial wings. That means VG plus is the dominant allele for wild type. And remember, in this case, wild type means normal wing. So there's three possible genotypes. You could have a fruit fly that's VG plus, VG plus. I know that sounds weird, but that's what it is. VG plus, VG plus. That's homozygous dominant for normal wing. Or you could have a heterozygous fly that's VG plus VG. That would be heterozygous. It would still have normal wings, though, because VG plus is dominant. And then the only way you're going to have a fly that looks like this with vestigial wings is if it's homozygous recessive, VG, VG. So it takes a little getting used to. I really don't like this system, and I really hate that I'm having to subject you to it because it is a little confusing, but this is just how you do it. And if you ever take a college biology class or a college genetics class, you're going to have to know this. And so I might as and you're going to see it on the AP exam. So I know it's weird, but you'll get used to it. All right, so as I said, T.H. Morgan was very interested in identifying new mutations in fruit fly populations and then trying to determine whether that mutation is a dominant allele or a recessive allele. Eventually, he was trying to figure out if certain traits were sex-linked or autosomal. And so, he was doing a lot of the same kinds of things with fruit flies that, that Mendel kind of was doing with pea plants, and really taking it even a step further than Mendel ever did. I already mentioned the vestigial wing mutation that Morgan was interested in, but one of Morgan's most famous contributions to genetics revolved around a mutation in fruit flies that causes the eye color to be white. So if you bear with me here, let me go back to, well, I guess right here. This is the typical eye color that we see in most fruit flies. It's kind of a bright red eye color. That's the wild type, red eyes. So Morgan was just astounded when he finally found a couple of flies that had a different eye color. It was white. And he said, well, that's got to be a mutation. He ended up calling it the white eye mutation for obvious reasons. And he used a lowercase w to represent that particular mutation. And then he set out to do some experiments to kind of figure out what sort of inheritance pattern he was dealing with. So in the parental generation, he crossed white-eyed males. These were true breeding, so they were white-eyed males that were, homozygous, so they came from a long line of nothing but white-eyed flies, and he mated those with wild-type females, meaning they were homozygous for red eyes. And if you do that, you could pause the video if you'd like and work it out on a Punnett square, but what it would predict is that in the F1 generation, we should see all red eyes. That's assuming that red eyes is dominant and white eyes is recessive, which that turns out to be exactly right. The red eye allele is, in fact, the dominant allele. And so it was no surprise that that's exactly what Morgan got. All of the F1 flies did have red eyes. And so if you kind of set this up, now just ignore the right side of the screen, look at the left side of the screen. If we set this up as an autosomal trait, here is the, what was it, white-eyed males. Okay, so here's the white-eye male, WW, homozygous white, crossed with red-eyed female, homozygous red, W plus is red, W is white. So this is homozygous white eyes crossed with homozygous red eyes. And of course, you're going to get nothing but heterozygous, W plus W. And because W plus is dominant, every one of these flies is going to have red eyes. And that matches the actual result that Morgan got. But how do we know this is not a sex-linked trait? You know, how do we know that the gene for white eyes is not carried on the X chromosome? What would that look like? Well, if it's a sex-linked trait, then that would mean that these males have W, which is the white eye allele, on their X chromosome. So here's a male crossed with homozygous red-eyed female, but you would get the same result, right? This is going to give me nothing but red-eyed offspring. So it's impossible to tell at this point, just by looking at the F1 results, it's impossible to tell if white eyes is an autosomal trait or is it a sex-linked trait. So it was the next generation of data that told the story, because now what he did was he took some of these red-eyed offspring and he bred them together. So he's taking a F1 red eye and he's breeding it with another F1 red eye, and let's see what color eyes we get in the F2 generation. So if this trait is autosomal, you would expect to get 3/4 red eyes in the F2 generation and 1/4 white eyes. And it should be a 50-50 split male-female, because male-female shouldn't have anything to do with eye color, right? So that should always be a 50-50 split. So let's look at why that is. So here's heterozygous red crossed with heterozygous red. So we get homozygous red, heterozygous red, heterozygous red. That's 3/4 red eyes and only 1/4 white eye. But it shouldn't matter about male-female. All of these should be, you know, half of them male, half of them female. And that does not square with the actual result that Morgan got. Because even though the flies did turn out 3/4 red eye and 1/4 white, he noticed something very peculiar. In the F2 generation, the only flies that had white eyes were male. And none of the white-eyed flies in this generation were female. Could this be a sex-linked trait? Well, let's see. If it's a sex-linked trait, here's our F1 red-eyed male. Here's a F1 red-eyed female. Now remember, she's heterozygous because this is the F1 generation. And so here we would get red-eyed female, red-eyed female, red-eyed male, and white-eyed male. So 3/4 of the baby flies should have red eyes. That's consistent with what Morgan got. 1/4 of the baby flies should have white eyes. That is also consistent with what Morgan got. But this predicts that all of the white-eyed flies will be male because they'll have a Y chromosome. See? And it predicts that 1/3 of the red-eyed flies will be male, and 2/3 of the red-eyed flies will be female. And it turns out this is the result that matches up perfectly with what T.H. Morgan actually got. He actually got exactly this: 3/4 red eyes, 1/4 white, but those were all male, not a single one of them female. And as far as the red-eyed offspring, 1/3 of them were male and 2/3 of them were female. So this was a big dramatic moment because for the first time in human history, a geneticist was able to definitively say that a particular gene for a particular trait is definitely carried on a particular chromosome. In this case, it's the white eye allele that is definitely carried on the X chromosome. Therefore, white eyes in fruit flies is sex-linked. It is carried only on the X chromosome. Now, in addition to this famous discovery of a sex-linked trait in Drosophila, Morgan also became very interested in a phenomenon which he referred to as linked genes. Now, this is different from sex-linked. This is just genes, different genes that are linked to each other. And what that means when we say genes are linked, it means that genes for different traits are on the same chromosome. For example, you may have a chromosome that has maybe the vestigial wing gene, but somewhere on that same chromosome, maybe there's a gene that affects body color. Or maybe there's a gene that affects the length of the antenna, or something like that. So whenever you have genes that are linked, that are located on the same chromosome, they are not going to assort independently. I know that's a little confusing, but that just means they're not going to follow Mendel's law of independent assortment. And whenever you have data, genetic data, that suggests that two genes are not following Mendel's law of independent assortment, it means that those two genes are linked. They are located on the same chromosome. And if they're really close together on the same chromosome, then they're definitely not going to assort independently. So we're going to be talking, I'm really just kind of planting a seed in your brain at this point. We're going to be talking a lot about linked genes, and we're going to be learning how we can use this understanding of linked genes to construct detailed maps of gene locations on chromosomes. So I'm not going to get into a lot of detail on this right now. I'm really just kind of planting a seed in your brain for later. Hopefully this seed will blossom into understanding later. Okay, just to recap here, the concept of linked genes, which we will discuss in much more detail later. Linked genes are genes for different traits that just happen to be carried on the same chromosome. They do not obey Mendel's law of independent assortment. However, they can assort independently due to crossing over. Crossing over is when homologous chromosomes break off pieces of DNA and swap with each other. Remember, that happens in prophase 1 of meiosis. So if that breaking off point where the switch happens between two chromosomes, if that breaking point occurs between the two linked genes, then they will assort independently. They will follow the law of independent assortment. And Morgan was a pretty smart guy. He figured out that this is a useful way to determine how close together linked genes are. If they're sitting on the same chromosome, are they really, really close together? Or are they spaced out on opposite ends of that chromosome? And so just to kind of illustrate this, I drew a picture of linked genes. This is the B represents a gene for body color. And you already know the VG represents wing shape. VG plus for normal wing and VG for vestigial wing. And like I said, B is for body color, and B plus is the normal gray. It's really more of a tan, but they call it gray body color. And B is the recessive mutation for black body, really, really, really dark colored body. And we know those genes are linked. Morgan was able to figure out that those are linked genes. They are physically located on the same chromosome. But what he wanted to know is, are they spaced out? Is there a lot of space in between their locations? The fancy word for location on a chromosome is locus, or loci is the plural. So are these two loci, these two gene locations, are they spaced out far apart like this, or are they really close together? So I drew it both ways, and Morgan figured out there's a way to tell, even though with early 1900 technology, you couldn't see the chromosomes really. You certainly couldn't see the genes on the chromosomes. And so there was no way to just look and see how spaced out these genes were. But he figured out a way to do it based on calculating what percent of the time a crossover event occurs in between the two genes. In other words, if a piece of this blue chromosome is going to break off and a piece of this red chromosome is going to break off, and they're going to switch places, crossing over, you can figure out whether that break happens in between the two genes, or does that break happen somewhere outside of the two genes? Like if the break happens here, see, it's not going to happen in between the two genes. But if the genes are spaced out and the break happens here and here, then it is happening in between the two genes. And you can actually calculate, I'm going to teach you how to do this, you can calculate what percent of the time the crossover break happens in between two genes. And if that turns out to be a high percent, then that means the genes are spaced really far apart. If, however, that percentage calculation turns out to be a low number, we call that low percent recombinance, that simply means that the genes are really, really close together. And so it's very unlikely that the crossover break would happen in between them. See, crossover is random. Where that break happens so that the chromosomes can swap DNA, it's totally random where that happens. So if the genes happen to be really, really close together without much space in between, it's going to be just pretty unlikely that a random break is going to happen in between them. But if they're far apart, then it's a lot more likely that that random break is going to happen in between them. So I know that's confusing. I mean, that probably doesn't even begin to make sense yet. But trust me, it will. We will make sense of this real soon. Okay, so what I'm sort of hinting at is a technique that was devised by Thomas Hunt Morgan, where genes, the locations of genes can be mapped. So he developed a way to draw the chromosome and actually figure out where the genes are located on the chromosome This is called a gene map, and it's based on this percentage calculation that I was just talking about, calculating percent recombinance. Sometimes it's called recombination frequency, and it's actually very easy to do this with a calculator. You just need some genetics data. You need some dihybrid cross actual data from fruit fly breeding. And so I'm going to give you some sample data. We're going to learn how to do these calculations. But what you're going to calculate is a value called percent recombinance. It's called percent recombinance or recombination frequency. And if it's a big number, that means the two genes in question are far apart. And if it's a small number, that means those two genes are close together. And so we can begin to construct a map that shows the relative positions of those genes on the chromosome. And so that's a good place to stop, but I do want to mention the extra credit phrase that pays. And this is a little different this time. This is an actual quote from a letter. I think it was a letter that Thomas Hunt Morgan wrote, or maybe an article. Sometime in the early 1900s, he was beginning his research on fruit flies, and he needed to identify a mutation so that he could study the inheritance of that mutation. And, with Gregor Mendel and his pea plants, that was super easy. If he wanted to order some purple flowered pea plant seeds, he just ordered them from the seed catalog. If he wanted white flowers, he would just order that. If he wanted tall plant, he could order that from the seed catalog. It was real easy. But for Morgan, he had to find his mutations. So he spent 2 years looking at hundreds of thousands of fruit flies under a microscope until he finally found a white-eyed fruit fly, because most of them had red eyes, and it took him 2 years to find one with a different eye color. And so right before he found that famous white eye mutation, he was quoted as saying this, and you can email me this phrase for your extra credit. Top 10, the first 10 to email me this get extra credit. Morgan said, 2 years work wasted. I have been breeding those flies for all that time, and I've got nothing out of it. 2 years work wasted. I have been breeding those flies for all that time, and I've got nothing out of it. You can tell he was pretty exasperated, but his hard work paid off, and he is considered to be one of the most important geneticists of all time. And that is the end of our lesson. Take care. I'll see you soon.